Welcome to Roadmap to Residency Evidence-Based Applications. Hello everyone, I am Abhiraz. Welcome back. In this video, we will be discussing about DNA synthesis, DNA replication, and DNA repair mechanisms. So DNA replication occurs during the S phase of interphase, and exact copies of all the DNA uh, on all the chromosomes are made, and this creates a sister chromatids, chromosomes and its copy, which will be separated in mitosis. Now, when the cell divides in mitosis, its daughter cell gets a full set of DNA, and when the cell divides, that is cytokinesis, there is a full set of chromosomes at each pole forming new nuclei. So that's why DNA replication is important. So DNA replication at a standard level. So number one is the enzyme DNA helicase. So DNA helicase, what does it do? That it unwinds and unzips the DNA. You can see that DNA here is winded and it is a zipped, that is tight. So what DNA helicase does is that it breaks the hydrogen bonds and it unwinds and unzips the DNA. That is the main role of DNA helicase enzyme. And this is the parent strand, and this one is also a parent strand, and these are the bases of the DNA which are exposed. Second up is the DNA polymerase, which creates complementary strands. Okay, and we can see that here there were the parent strands, and what DNA polymerase do is that it creates a complementary strands on both sides. Okay, so DNA polymerase moves in opposite directions on each strand and makes up a complementary strand. The free nucleotides are collected by DNA polymerase and are attached to the new strand by complementary base pairing. Okay, now, the complementary base pairing ensures the identical copies of DNA. Now, the parent strands act as a template for the new or complementary strands. The adenine pairs only with thymine. The cytosine pairs only with guanine. And this ensures the new DNA molecules is identical to the original DNA molecule. So the no mistakes are made. So the base sequences of nucleotides is conserved, and we can again practice this ourselves to learn genetics, and we can just practice among. So there is A, there is C, so where will C act with, where will A act with, so you just need to practice this all thing. Okay, so it's important that there are no mistakes in the order of the bases, and mistakes could be fatal to the organism. So DNA replication is semi-conservative process, that the new strands contain one original strand, that is one parent strand, and one copy, that is the complementary strand. So that's why it is a semi-conservative process. We conserve one strand of the DNA. We do not completely form a new DNA. So DNA replication is initiated at many points in eukaryotic DNA, and this makes the DNA replication faster and more efficient. These points are known as the origins of initiation and will have the same or pretty similar base sequences. The proteins are called as origin recognition complexes, will bind here, and then DNA's helicase will be able to attach to begin replication. The replication forks will move along the DNA strand in the same direction. The replication in prokaryotes is bidirectional and initiated from a single origin because prokaryote DNA is looped, but eukaryotes have long strand. Other processes are essentially the same. So the DNA replication moves in 5' to 3' direction. This means the replication starts at the 5' and end at the 5' end of the new strand. Okay. Now DNA, this means that replication, the 5' end of the next nucleotide attaches to the 3' carbon of the last one to join the new strand. Okay. So 5' and again there is the last one is the 3'. Again, now, this means replication starts at the 5' end of the new strand. Replication on the lead strand is continuous. Okay, so this is the lead strand or the parent strand is continuous. Okay, and replication on the lag strand, there is leapfrogs. Okay, so there was a lead strand, and this is the lagging strand. Yeah. So 3' to 5' is the lead strand. It moves from 5' direction to 3'. For the new strand, it is 3', and for the new strand, it is 5'. But the another end of the DNA is called the lagging strand, where there is leapfrogs, there is breaks. Okay. The hard part is the lag strand replication. This is more complex because the DNA polymerase is moving in the opposite direction to the DNA helicase. Okay, now DNA helicase is moving in this direction. DNA polymerase is moving in the same direction in the lead strand. But in the lagging strand, the DNA polymerase moves in the opposite direction. So that's why it is very hard and complex. The RNA primers are used to mark position, and replication goes in a leapfrog style from section to section. One section, another section, and at last, these all sections join together. So DNA helicase unwinds and unzips the base pair. In prokaryotes too, DNA polymerase makes a complementary strand on the leading strand adding nucleotides to the 3' end of the complementary new strand. The RNA primase follows the helicase, leaving RNA primers, and these are markers for initiation of DNA polymerase on the lag strands. And the DNA polymerase attaches to an RNA primer and replicates the DNA in a 5' to 3' direction. Now, when it reaches the another RNA primer, it detaches and leapfrogs to the next primer following the helicase, right? And these are called as the Okazaki fragments. So our DNA polymerase 1 moves along the replication fork, removing the RNA primers, and DNA ligase attaches the Okazaki fragments into a continuous strand of DNA. You know that there is one strand, another strand in the lagging side. Now, what happens is that DNA polymerase will remove these primers, and DNA ligase what will do? Ligase will again bind all this. So ligase means binding. So the enzymes and molecules of DNA replication are helicase, which unwinds the DNA and breaks the H bonds or hydrogen bonds between the base pairs. DNA polymerase 3, which attaches the nucleotides in the 5' to 3' direction. RNA polymerase, which leaves the RNA primers on the lag strand. RNA primers, which are the initiation site for DNA polymerase 3 on the lag strand. Okazaki fragments, sections of a new DNA on the lag strand. DNA polymerase 1 removes the RNA primers. DNA ligase attaches the Okazaki fragments together. So DNA replication is that it occurs in 5' to 3' direction in continuous and discontinuous fashion. It is a semi-conservative process, and it is more complex in eukaryotes than in prokaryotes, but shares analogous enzyme. So here is the part from the first aid only. So origin of replication, that is the particular consensus sequence in genome where DNA replication begins, maybe single or multiple. We have talked about it. There is formation of a Y-shaped replication fork. So this is a Y-shaped replication fork, right? So here we can see this is the origin of replication, right? And this is the Y-shaped replication fork, right? Here we can see the enzyme helicase, which is unwinding the DNA. Then we can see the single-stranded binding protein here, which makes sure that the DNA remains unwound. The DNA does not again go and attach together. And again, there is the primase, okay? And again, then after that, we have the okay, so after that we have primase. We go up with the replication by DNA polymerase. Okay, and again DNA polymerase brings the nucleotides, and after that we have the DNA polymerase 1 in the lagging strand, and like that again after that we have the DNA polymerase, or sorry, DNA ligase, which joins the lagging strand. This is the whole summary of everything. Talking about telomerase, which is present in eukaryotes only, it is a reverse transcriptase that adds the DNA to the 3' end of the chromosomes to avoid loss of genetic material with every duplication. Upregulated in progenitor cells and also often in cancer. Downregulated in aging and progeria. Here we have one question just for your recall. A 45-year-old man presents to the emergency department with symptoms of chest pain and shortness of breath. An electrocardiogram reveals ST segment elevation in the inferior leads. He is diagnosed with an acute myocardial infarction. Genetic testing reveals a mutation in an enzyme responsible for unwinding the DNA double helix. You know what is the answer? The answer is DNA helicase, option number C. Now, as we have talked that there should be perfect replication, there should be not be any mistake. Otherwise, it can lead to a fatal manifestation in the human being. So that's why there are mechanisms for DNA repair. So for the double strand repair, we have the non-homologous end joining and homologous combination. So non-homologous end joining is that it brings together two ends of DNA fragments to repair the double-stranded breaks. The homology is not required. Part of the DNA may be lost or translocated. For example, if this part of the DNA is lost or translocated, then what non-homologous end joining will do is that it will join these two break in the DNA. Simple as that. But it may be dysfunctional in case of ataxia-telangiectasia. You need to remember these clinical syndromes. Homologous recombination is when it requires two homologous DNA duplexes. A strand from the damaged double-stranded DNA is repaired using a complementary strand from intact homologous double-stranded DNA as a template. It is defective in breakup mutations and in Fanconi anemia. Another is a single strand repair pathway. So the number one is the nucleotide excision repair, where the excessive endonucleases remove the oligonucleotides containing damaged bases. So if there are damaged bases, they will remove these bases. This is called as nucleotide excision. They just excise the damaged base, and it is defective in xeroderma pigmentosa. In case of base excision repair, the base-specific glycosylase removes the altered base. If there is only one base defect, then there is a specific enzyme called as glycosylase, and the name is important because they will ask you the name. and that will come and that will just excise that defective base. I'm talking about the mismatched repair. The mismatched nucleotides and newly synthesized strands are removed and gap is filled and resealed. For example, there is a very mismatched configuration there. Then what will happen is that whole part is will be removed and the gap will be again filled and it will be again resealed by the DNA ligase enzyme. And it is defective in case of Lynch syndrome or hereditary non-polyposis colorectal cancer. So 55-year-old woman presents to her oncologist with a history of breast cancer. She underwent chemotherapy and radiation therapy 5 years ago. Recently, she developed a new tumor at the site of previous radiation. Genetic testing reveals a mutation in enzyme responsible for DNA repair mechanism, what may be most likely affected. So the correct is non-homologous end joining repair, which is a mechanism used by cells to repair double-stranded breaks. It involves the direct rejoining of the broken DNA ends without the requirement of for extensive homology between the broken ends. In this patient, the development of a new tumor at the site of previous radiation therapy suggests that the tumor cells may have accumulated additional DNA damage due to the radiation exposure. So what I would suggest you is that in case of DNA synthesis, RNA synthesis, DNA repair, please go through your first step, go through these videos again and again, and then you will be uh, just a perfect or near perfect in answering these sort of questions. So this require understanding, and for these sort of topics, instead of solving more and more questions, I will suggest you to read more and more the same thing and stick it to your mind, because they can ask you anything, any name, any enzyme, any disease, any clinical association. Thank you for watching.